Eversion takes place between the moment of the pupae and 4 hours after pupae. And afterwards, the dorsal and the ventral epithelium are in contact with each other, and wing is an epithelium with 2 layers that starts elongating and increasingly having a shape that resembles the adult wing shape. So this growing process takes place between 4 and 8 hours after pup, more or less. And you can see pictures here. So in the first place, between 4 and 6 hours after pupae, the width of the wing or the head decreases, as you can see from the blue arrows. So the wing becomes narrower, but it elongates. It becomes longer in the proximal-distal direction, as you can see from the yellow arrow. After 6 hours after pupae, this change in shape stops and the wing increases its size isotropically, which means that both its width or head and its length increase at the same rate, more or less. Here you can see videos. In the upper part, there's this change in shape between 4 and 6 hours, and in the lower part you can see the increase in cell area, in wing area of the second part. These changes can be measured. So in the upper left part, the width is plotted for different times, at different times. So you can see that between 4 and 5 hours the width of the wing decreases, and then between 5 and 7 hours it increases. However, the length of the wing increases constantly between 4 and 7 hours. So if you calculate some shape index, which is length minus height of the wing, you can see how this shape changes sharply between 4 and 5 hours after pupae, but then it remains more or less constant or even decreases a little between 5 and 7 hours after pupae. On the other hand, wing area stays constant between 4 and 6 hours and then increases between 6 and 7 hours. So in this region here, between 4 and 6 hours, the shape of the wing is changing, but its area is not. Whereas in the last part, the opposite happens. The area is increasing, whereas the shape is mostly not changing. There are 2 separate parts, therefore, in this growing process. The first one can be regarded as a convergent extension process, because the tissue becomes narrower and more elongated, whereas the second part can be regarded as a simple expansion process. Different cell behaviors can be responsible for these changes. For instance, cell division, cell death, changes in tissue topology or the neighboring relationships between cells, and of course changes in cell shape and cell size. Here wings are stained for phosphohistone 3 in red, which marks cell division, and as you can see there's not a lot of cell division between 4 and 8 hours after pupae, so probably cell division is not very important for determining or driving these changes. On the other hand, you can see here cell death. So actually what has been measured is cell extrusion. This central cell is suffering a lot of pressure from their surrounding neighbors, so it gets smaller and smaller, and it is finally extruded from the tissue, and it dies probably, but some not necessarily. So here you have pictures where the central white cell is being extruded, and there you have a video where, well, here I'm going to put it again. Here you have a cell that is being extruded. As you can see, it gets smaller. Also, T1 transitions is how topological changes are, or change of neighbor are called. This is, well, basically when you have one cell junction, it is replaced by a cell junction in the opposite direction. So at the beginning, these two white cells are in contact with each other, and then they lose contact and the two gray cells become in contact. So this part of the tissue is more elongated in the vertical direction at the beginning, and at the end this region of the tissue is more elongated in the horizontal direction. Therefore, you can see how these topological changes result in shape changes. So in the video in the left, in the right, you can see one of these transitions taking place here. One cell junction that has just formed and is elongated. Okay, so then changes in cell shape can also result in changes in tissue shape, of course. Here you see that in the wing, cells are elongated in a vertical or anterior-posterior direction at the beginning, and then they become less elongated. So this decrease in anterior-posterior elongation results in more proximo-distal elongation. And finally, changes in cell shape can also result in, well, in changes, of course, in cell area, can also result in changes in the area of the tissue. Here, cells are simply increasing their size. Okay, so you can see how they are small and they become bigger in the video. They clearly increase their apical size. So we can quantify all of these behaviors and measure what's their contribution to the overall changes in tissue shape and size. In the F figure, you can see how rearrangement or T1 transition, neighbor exchange between cells, takes place mostly between 4 and 5 hours after the pupae, whereas it doesn't happen a lot between 6 and 7 hours after pupae. So this change in cell neighbors is responsible or maybe contribute to change in shape of the tissue. Then changes in cell shape. As you can see here, they measure the shape of the cell or some elongation index. And as you can see, cells are higher, or they have a higher height at the beginning at 4 hours, and they become less anterior-posteriorly elongated. Okay, so this is also contributing to the overall change in shape. Finally, this is change in apical area, and as you can see, the change in area in cell area increases towards the end of the process, towards between 6 and 7 hours after pupae. However, even if you are measuring these changes in cell area, maybe there's something else that you are missing. So that's why they plot this thing here. So they plot in the x-axis the increase in overall tissue area, and in the y-axis the increase in cell area, the contributions of individual cells. And as you can see, the correlation is very high, so most of the change in cell area during this last part is determined by most of the change in tissue area is determined by changes in cell area and not by any other mechanism. And additionally, here's some kind of prediction or some kind of measurement of what is the contribution of rearrangements to the overall sharing of the tissue. And as you can see, this contribution is high at 4 hours after pupae, but then it becomes lower. So what molecular mechanisms are responsible of these changes in shape and size? First, I'm going to talk about the molecular changes in the expansion, and then about the ones during convergent extension, which is the opposite order, the reverse order, but I thought it would be easier to tell it this way. So during the expansion, between 6 and 8 hours after pupae, cells change their shape from columnar to cuboidal. So in the beginning you have these cells that are very tall, and the tissue where goes convergent extension, and they are still very tall, and then they just become flatter. So as a result, their apical area becomes bigger, so they increase their area, and isotropic growth takes place. So why do these cells where go this transition, this columnar to cuboidal transition? Well, during this time there's an degradation of extracellular matrix, both the apical one and the basal one. So apical matrix and basal matrix have different components. The basal extracellular matrix has collagen 4. One of its components is Viking, or one of the subunits is Viking, and the apical matrix has DAMPI. Okay, so different proteins are degraded by different proteases. And you can see here in green the apical extracellular matrix, and in the lower part the basal extracellular matrix, and you can see how it is degraded between 4 and 7 hours after pupae, and it remains there between 0 and 4 hours. And here's the quantification of the percentage of the epithelium that is covered by this extracellular matrix, and as you can see, it decreases sharply between 4 and 5 hours after pupae. So if these proteases are repressed, like Stabile, or RNAi against Stabile, or these protease inhibitors, the wing is unable to elongate. Here in the top you have a control, okay, and in the lower part you have this inhibition, and you have so the green is the apical matrix, which is marked with DAMPI. It's a DAMPI reporter, and as you can see, if it is not degraded, the wing cannot elongate. So the same happens when you prevent the degradation of the basal matrix. Here in green you have a Viking reporter, which, as I said, is one of the subunits of collagen 4. And if you add a protease inhibitor, or you overexpress TIMP, which is a repressor of the proteases that degrade collagen, in either case you have that the basal matrix is not degraded, as you can see it here in the inner part of the disc, and the wing does not elongate. Okay, it doesn't. The expansion process does not take place. And as this would be the result in an adult wing that, as you can see, it cannot grow. Regarding molecular changes during convergent extension, it has been shown that myosin II is planar polarized at 4 hours after pupae along the anterior-posterior axis. So here you can see in green SQH, GFP, which is the heavy chain of myosin II, and you can see how it is preferentially located in anterior-posterior cell junctions, the vertical ones. At 8 hours after pupae, this is not the case anymore. So you can see as how myosin 2 is not in these junctions anymore. It is not only that it stops being polarized, but it also stops being in the apical part of the cell. Here you can see a lateral view of this epithelium, and at 4 hours after pupae, myosin 2 is located mostly in the apical side of the cell, whereas at 8 hours it is located in the more basal or lateral, in the lateral region of the cells, as these schemes, these drawings also show. Here you can see like a view of the whole wing. These vertical lines mark how myosin 2 is polarized at the beginning, and then it stops being polarized, and then you cannot even see it in the apical side of the cells. Here you have a quantification along with several pictures of this process of changes in myosin 2 location. As you can see here in the central places, myosin 2 is located at these anterior-posterior junctions mostly, and in the apical side, and slowly it becomes located in the lateral side of the cells, and it is not anymore polarized when you look wing from the frontal view. And well, here's the more precise quantification. If myosin 2 is inhibited by, the Rho kinase is pharmacologically inhibited, and Rho kinase is responsible for activating myosin 2, you can see how the wing fails to elongate, as in the lower picture. The one in the top would be a control. Okay, so this convergent extension process does not take place if myosin 2 cannot contract. So what is happening there really, and why does myosin 2 stop being polarized? Well, what has been proposed is that after the extracellular matrix degradation, these columnar cells become more relaxed. Okay, it's the effect like if you have a balloon in your hand and you're pressing it and then you release your hand, so the balloon becomes more rounded because it is a shape that is energetically more favorable. So this is what happens with cells. They just, after removal of the extracellular matrix, they become more rounded, they become more cuboidal, so their basal and apical sides expand and increase their area. Okay, with this area expansion, the cell-to-cell junctions, these adherens junctions, are released, and the actomyosin ring in the apical side is also disappears. It is dispersed. So myosin disappears this way from the apical side, and later it is going to be recruited in the lateral side. It is going to be recruited there, so it will be exerting some tension again and helping the cells to become flatter, helping the epithelium to become flatter and more cuboidal. So as a summary, the process of elongation of the wing blade, which takes place between 4 hours and 8 hours after pupae, takes place in two phases. The first one is a process of convergent extension, where there is elongation of the tissue but the area remains constant, and the second phase is an expansion, an isotropic growth, which in which the shape does not change but the area increases. So cell changes in cell shape and in cell contact topology explain this convergent extension, and the changes in cell area, the aggregation of changes in the area of all cells, explain the expansion of the tissue. So these are the cell behaviors that explain this process. Then convergent extension is dependent on myosin 2 polarization, and expansion is the result of columnar to cuboidal transition. The extracellular matrix degradation allows these changes to happen, and this extracellular matrix degradation is triggered by the peak of ECD zone, by which the beginning of the pupae stage is regulated. Finally, this process, or this stage, is not thought to contribute a lot to variation in wing shape in adults. However, it is possible, or it is plausible, that the relative amount or the relative time that wings spend elongating or expanding could affect the shape of the wing. So changes in the timing of expression of proteases, as well as changes in the establishment of MYO2 polarization, could possibly mediate these changes, so that wings would become more or less elongated, or even more or less big, if this timing is altered. Finally, I would like to make some evolutionary comment, regarding the evolution of halters. As we have already mentioned, other insects, such as odonates, have 4 wings, and the rear wing has been lost in Drosophila, but this appendage, the halter, remains there and is considered to be a homologue of the wing, of the second pair of wings in other insects. Some mutants, typically ultravithorax mutants, have a duplication of the second thoracic segment, and this includes having 2 extra wings. So it has been shown that halters do not remodel their extracellular matrix between 4 and 7 hours after pupae. Here you can see the perlecan, which is a component of the apical matrix, and laminin B at 4 hours and at 7 hours after pupae, and they are not removed in the halter. However, in the leg they are removed, and this allows also the leg to expand, and in the wing, as we have already discussed. So here you can see the quantification, how this is the amount of laminin B and perlecan at 4 and at 7 hours after pupae. So in the wing decreases, in the leg decreases, and in the halter it doesn't. So UBX has been shown to repress stable, which is the protein that degrades DAMPI in the halter. So if you have this is stable expression in the halter, and as you can see, it is not expressed at all. However, if you repress ultravithorax with RNAi, this stable expression increases, and as you can see, at 7 hours this results in a slight increase in the elongation of the halter. Additionally, it has been shown that by ChIP-seq that UBX binds directly to these protease regulatory regions. It binds to stable here, it binds to notopleural, which is another protease which I haven't mentioned, but it also degrades DAMPI. Here in these red peaks is where ultravithorax binds and represses them in the halter, and then ultravithorax binds to MMP2 and MMP1, which are proteases that degrade the apical the basal matrix, which has collagen, and also ultravithorax binds to TIMP gene, but in this case ultravithorax is not repressing it, it's activating it, and TIM is also a repressor of MMP1 and MMP2. So this is the model that the authors propose. Basically, this thoracic segment 3 is always in all insects is expressing ultravithorax, but the wing genes are not sensitive to ultravithorax repression, so they have wings. Then in Drosophila, ultravithorax is repressing wing genes and activating some halter genes because wing genes have become sensitive to ultravithorax. So it is repressing stable and notopleural, and ultravithorax is activating TIMP and in turn repressing MMP1 and MMP2, therefore preventing the extracellular matrix degradation, which does not allow the halter to develop into a wing because it's mechanically constraining it. Okay. However, when you mutate ultravithorax, this is not happening and you have wing elongation like in other insects. Of course, ultravithorax is doing more stuff, but this is just one of the things that one of the ways that it has to select for the fate of the thoracic segment 3.